Hi, and welcome to our video presentation in exploring protein in its structure, folding, and many more. Proteins are incredibly important because that is what we are made of. When you look at the mirror, you are looking at your own protein. So we can find proteins everywhere, especially in our foods, such as the milk, which is considered to be a complete protein, from the chicken, a high in protein food, the almond, the fish, and the egg.
Proteins, a naturally occurring unbranched polymer in which the monomer units are amino acids. Maintenance of body tissue, major source of energy, involved in the creation of some hormones, help control on body function that involve the interaction of several organs and helps regulate cell growth. Protein folding, process by which a polypeptide chain folds to become a biologically active protein on its native 3D structure. Folded proteins are held together by various molecular interactions. Each protein is synthesized as a linear chain of amino acids or a random coil which does not have a stable 3D structure. Protein folding occurs in a cellular compartment called the endoplasmic reticulum. This is a vital cellular process because proteins must be correctly folded into specific three-dimensional shapes in order to function correctly. Unfolded or misfolded proteins contribute to the pathology of many diseases.
When we talk about protein folding, we need to understand different levels of protein structure because there are different ways of folding that can happen in the different structural levels. The four levels of protein structure are primary, secondary, tertiary, and quaternary. It is helpful to understand the nature and function of each level of protein structure in order to fully understand how a protein works. The shape of a protein is critical to its function because it determines whether the protein can interact with other molecules. First level of protein structure is primary structure. This is the sequence of amino acids that make up a protein. Amino acids are the monomer, which means the building block of a protein. They are held together by a peptide bonds. In protein synthesis, amino acids are added to form polypeptide chain and proteins are made of one or more of these polypeptide chains. In short, primary structure describes the unique order in which amino acids are linked together to form a protein. The next level of protein structure is the secondary structure. The secondary structure is the sequence of amino acids from the primary structure can be folded in different ways. The most common ways are the alpha helix and the beta pleated sheet. And one of these foldings, the protein does depend on the amino acid arrangement it has. Both of these shapes are due largely in a part to hydrogen bonds. Hydrogen bonds can occur specific areas of the protein's amino acid. The third level of protein structure is the tertiary structure. Tertiary structure is looking at more folding that occurs 3D shape of a functional protein. And lot of this is due because of the R groups, also called side chain. The amino group and the carboxyl group are generally standard parts of an amino acid, although the R groups found in amino acids can vary among different amino acids. There are several types of bonds and forces that hold a protein in its tertiary structure. First is the hydrophobic interactions, next is the hydrogen bonding, and last is the ionic bonding. Last level of protein folding is the quaternary structure, where a protein consists of more than one polypeptide chain. Each of these polypeptide chains can be a subunit, and interactions between them such as hydrogen bonds and disulfide bonds can keep them together. They may also be composed of different subunits. Hemoglobin is an example of a protein with quaternary structure. Hemoglobin, found in the blood, is an iron-containing protein that binds oxygen molecules.
So, there are factors that affecting the protein folding. In the next slide, we are going to talk about it and explore more. So, protein folding is a very sensitive process that is influenced by several external factors including electric and magnetic fields, temperature, pH, chemicals, space limitation, and molecular crowding. Extreme temperatures affect the stability of proteins and cause them to unfold or denature. Similarly, extreme pH, mechanical forces, and chemical denaturants can denature protein.
What are the intermolecular forces involved in protein structure? We have 6 major intermolecular forces that are involved in this process. First, we have the hydrogen bonding. It is a bond between sections of protein chains that are responsible for secondary structure of the proteins. Hydrogen bonding is very important in protein structure because it stabilizes the secondary, tertiary, and quaternary structure of proteins which formed by alpha helix between sheets, turns, and loops. Next, we have the dipole-dipole interaction, and it is also known as the van der Waals interaction. Dipole-dipole interactions are weak interactions that arise from the close association of permanent or induced dipoles. Collectively, these forces are known as the van der Waals interaction. Proteins that contains large numbers of this interaction varies considerably in strength. We have the peptide bonds, which links amino acid residues in a polypeptide that are formed in a condensation reaction between the acidic carboxyl group of one amino acid and the basic amino group of another amino acid. Next, we have the disulfide bonds. It stabilizes protein structure by organizing and destabilizing the denatured protein to the native structure. It also plays a critical stabilizing role in many protein structure because it forms a cross-links between different regions of polypeptide chains. Lastly, we have the hydrophobic bond. It is formed between two nonpolar groups or hydrophobic amino acid. It forms an interior hydrophobic protein core where most hydrophobic side chains can closely associate and are shielded from interaction with solvent water.
Sometimes proteins are misfolded, and misfolded proteins are not only a waste of energy for a cell, but they are also dangerous. These misfolded proteins typically have exposed hydrophobic regions, which can cause them to clump together, forming large aggregates. It can give disease such as sickle cell, mad cow disease, and Alzheimer's disease. Chaperones work by binding to expose hydrophobic patches on misfolded proteins or incompletely folded proteins by hydrolyzing ATP. The proteins that chaperones help fold properly are called client proteins. When the chaperones haven't succeeded fixing or haven't reached the misfolded proteins, the proteasomes will degrade it. Proteasomes destroys misfolded and damaged proteins through proteolysis.